Good afternoon, everyone. I'm Dr. Sridivya, and today we will be looking into the sewage analysis and treatment technologies. As in the previous class, we have learned about the filtration process, that is both rapid sand filtration and slow sand filtration units and all. So we have thoroughly known about this types of filtration units and what are its purposes and how it is like designed, what are its operational and phases. Everything we have discussed in the earlier class, and we have already started with the sewage analysis, but let me give you a quick recap of all these this topic, especially the sewage analysis. And the sewage is nothing but the wastewater which is mainly used, mainly coming from the municipal domestic purposes, etc. And this the importance, this sewage and wastewater, and these days particularly, these are not just considered as a waste, but it is morely considered as a resource, because with the ongoing water secures scarcity and water pollution, there is very less amount of water, fresh water resources available. And in order to make use of the resource, mainly this wastewater generation is also very high. And sewage as well. So sewage is nothing but it is consisting of more amount of nutrients present in it, which is not used for the domestic or portable purposes. So that is why it is often considered as waste, and if it cannot be utilized for any other purposes. But this, yes, this mainly has several important analysis in terms of public health, in terms of environmental protection, in terms of resource recovery. Sewage has special importance, and mm, it is mainly used as a public health. So this sewage, since it is consisting of several amount of pollutants, it is not considered as a resource for utilization, because it is carrying mainly for the, it carries main pathogens, including bacteria, viruses, parasites, which are like mainly the waterborne diseases. So for the public health, in concern of the public health, there should be like disease prevention. But this sewage carries more amount of pathogens and bacteria, with that causes the waterborne diseases. And analysis, which will actually help us to identify and quantify this pathogen, and timely interventions to prevent the outbreaks such as corona, like that kind of things, through water and all. It can be prevented by timely analysis and all. And by monitoring the substance use, that is the sewage analysis can also be used to monitor the presence of drugs and other substances of a community, providing the valuable insights into the public health trends, and also to ensure the safe water supplies, where the proper sewage treatment should be considered, and analysis are also is necessary for protecting the drinking water sources from the contamination point of view. And in terms of environmental protection also, the protecting the environmental ecosystems, this untreated sewage can deplete the oxygen levels in the rivers and lakes, harming the aquatic life. And excess nutrients such as nitrogen and phosphorus also causes these algal blooms, which further disrupt the aquatic ecosystems. And also the analysis, mainly it helps in monitor and control the discharge of pollutants into the environment. And to prevent the soil contamination, the disposal of sewage should, which mainly contributes to the soil contaminants, by consisting of harmful pollutants, and affecting the agricultural productivity and posing the risk to the human health and all. And to monitor the pollution levels also is mainly considered, because the sewage analysis allows the monitoring of different types of pollutions, from though it may be heavy metal contaminations to the presence of microplastics and all. So these are all the emerging pollutants which ultimately causes the sewage pollutions as well. And in terms of resource recovery, water reuse should be done, because the sewage can treated sewage can be reused for irrigation purposes. All right, sewage has to undergo like different technologies, that is the preliminary, primary, secondary, and tertiary level of treatments. And further, the treated sewage with zero pollutants and all can be reused for the irrigation purposes, industrial process, where industries can reuse this as a coolant, because mainly the water which can be used for the coolers and all, and also the even drinking water for the ROs and all, it can be used and also help to conserve the valuable resources. This water can be reused once it is treated in a appropriate manner by using the disinfectant process and all such things. And also in terms of nutrient recovery as well, these sewage consist of valuable nutrients such as the NPK, and which can be recovered and used as a fertilizers as well. So these recently the several researchers are focusing on the nutrient recovery process of several sludge, you can say, or the sewage, wastewater, everything. They are practicing this nutrient recovering technologies, such as the waste coming from from the environment should be recovered and then sending back into the environment in order to protect the environment as well. So in the form of fertilizers and all, we can reuse and introduce into the environmental as well. And in terms of energy production, so this energy, particularly this sludge which is produced through by sedimentation process or after the tertiary treatment and all, which can be used to produce the biogas by and which is a renewable energy source. So this green biogas is like mainly coming out from the sludge process, is like very much useful, and through this also like most of the hydrogen can also be developed and produced, and which is hydrogen is now the green gas which is like renewable source, which is mainly used for the automobile industries particularly these days, and it is very much upcoming. And this is mainly done by the anaerobic process, that and all will be looking into this in the later stages, and mainly used for the energy production as well, and valuable material recovery. And research is being done into recovering many different types of vulnerable materials from the sewage, including the precious metals as well. So coming to the characteristics and samplings, so mainly the physicochemical characteristics we have to look into, and the physical as in what we see and what we touch and what we smell and all. That is the touch as in the temperature, typically that higher than the ambient water due to its domestic and industrial discharges and the microbial activity going present in the water. And the color may ranges like difference from the gray water, that is fresh sewage will be there, to dark brown in color or the black color. And the odor like fresh sewage has a slightly musty odor, while septic sewage will be of all sulfurous smell due to the hydrogen sulfide production and all. And the turbidity, cloudiness caused by the mainly caused by the suspended solids present in the sewage water. And this total solids also, like total suspended or the dissolved solids, are the ones which are present in the water. And these are all the physical characteristics which determines the color, odor, amount of solids presence and all we will be understanding. While the chemical characteristics include the organic matter. This organic matter can be either calculated in a biological oxygen demand procedure or by the chemical oxygen demand. So this BOD is nothing but it measures the oxygen amount of oxygen consumed by the microorganisms by decomposing the organic matter. While the microorganisms take up the oxygen by while decomposing the organic matter, in the chemical oxygen demand, that is in COD, it measures the required total amount of oxygen which is required to oxidize the organic matter. So here the main role about the microorganisms, how much amount of O2 is consumed by the microorganism, that is how we will be knowing the degraded amount of organic matter. Here in COD, we will be measuring the amount of total O2 required, to oxidize the organic matter. So that and all will be discovered by the COD procedure. And nutrients, particularly the nitrogen and phosphorus, these are the main nutrients present and causes of eutrophication and leachate manner, and mainly present nitrogen mainly present as the organic nitrogen, ammonia, nitrite and nitrate, and phosphorus mainly present as phosphates form, and the pH typically it is alkaline, but it can also vary, and heavy metals mainly coming out from the industrial discharges, and the toxic organic compounds that is mainly coming from the pesticides and pharmaceuticals, and fog, mainly the fats, oil, and grease coming from the food processing and domestic waste, and the biological characteristics, which includes the pathogens and the indicator organisms. That is the presence of pathogens, that is the bacteria, virus, protozoa, helminths. All these are the pathogens which are included in the sewage samples, and the indicator organisms, that is the E. coli, that is Escherichia coli, and the fecal coliform, which mainly indicates the fecal contamination. That is, since the sewage also collects the domestic wastewater, toilet flush, and all these also will be collected together. So this is also like the presence of these indicator organisms also indicates the level of pollution of it. And the mainly the sampling techniques, mainly by the representative sampling, that is the samples should be accurately reflect the overall composition of the sewage. So the sampling should be done appropriately in order to reflect the composition of the overall sewage, and it may also involve like one time sampling or two times. No, it is not like that, but it is like completely throughout the year at a particular period, regular intervals of time, the sample should be taken, that will be collected and sent for the laboratory analysis. And this is how the year wise sampling should be taken at during the summer season, what will be the sampling rate, or what could be the sewage composition, and maybe during the rainy season there will be the dilution of water because of the rain, and sewage contaminants level will be slightly lesser, while the winter season, if it is too cold, the microorganisms activity will be very less, and therefore there will be little breakdown of organic matter, and there will be more pollutant load as well. So there are like different, due to the according to the seasonal variations and all, there will be a different type of pollutant level. So that is how like representative sampling should be done. And mainly like it has to be like taken multiple times in order to have the accuracy of the sewage pollutants. And the sampling will be done by composite samples or the grab samples. And the location of the sample point should be very precise. That is, the sampling point should be selected that represents the whole data. All right, so the location of the sample, so for example, if we are taking the sewage samples from the Hussein Sagar, be it Hussein Sagar or any drainage, there will be several points of it. So one in the middle, one at the adjacent of the shoreline, or maybe at like towards the right side, or maybe towards far here and there. There will be different selected sampling points, and which can represent the data of the whole lake and all. So they we should also consider the flow patterns, and also the potential sources of contamination. So where exactly the water from the industries or any pollutants which are getting coming, what are the inlet points, so the what will be the inlet points and all also will be for considering such kind of factors. The inlet point of solution pollution, the outlet part of the exit of water, that kind of, that can be in the another point. In the center there will be another point towards the adjacent or the shoreline. So there are several points and factors as well, like flow patterns also, which has to be considered for the sampling location. And the frequency of the sampling also depends upon the purpose of the analysis and the variability of the sewage. And sampling methods is of grab sampling, that is simple single samples taking at a specific period of time. But the composite samples are the mixture of grab samples taken over a period of time. That is like different seasonal variations, so different period of time it will be taken, but only graph sampling at one point of time only. Throughout the year only one time it will be taken. And then automatic samplers are used for the continuous monitoring, so there will be some probes or something where continuously now and then, so hourly wise also there will be maximum degrade changes in the BOD, hourly wise also there will be changes in the COD as well. So basically it depends upon the type of sampling, also depends upon the type of purpose. So depending upon the purpose of the project, or depending on the purpose of the evaluating the sewage contamination levels and all, it varies depending, and depending on the purpose the type of sampling also will be done. And the preservation techniques also are very crucial in order to prevent the changes in the sample composition. So once you don't preserve it in an appropriate manner, so there is no point of even taking the samples, because at the point of collection there will be a COD of maybe 1000 mg per gram per mg per gram per liter per day. And then again if you get it back to the laboratory and you don't store it appropriately, might be it will be less than to 750 mg per liter per gram. So the liter per day, that is how it can be reduced. So there it will be highly polluted. By the time you get it to the laboratory and you don't preserve it, at the end 1 or 2 days it will be lowered to 700, which is again there is a huge variation of it. So that is the reason why should be a proper preservation of the sample, such that the microalgae, so preferably it will be like a below 10 or below -4, like that they will be kept, such that or or maybe like under 10 only probably, because the activity of the microorganisms will be very less, and therefore the degrading properties of these organic matter also will be very less during that lower temperature. So the mainly the lower temperature inactivates the microorganisms activity. So that is how the preservation should be done, and mainly involve the refrigeration process or acidification process, or addition of the chemical preservatives, etc. And the handling of the sample. Since we have collected, we have preserved and all, the handling of the sewage is also very important, because it consists of heavy metals, pathogens and all these. And so proper mask should be worn, proper gloves, proper aprons, and everything should be worn. And then the containers, the use of proper appropriate containers also should be um, there for the usage of the storing these sewage samples. And then also proper labeling should be there, and properly handled in order to avoid the contamination. And there are several analytical techniques involved for this each and every analysis. For the physical analysis, the temperature probe is there, that is the thermometer or the electric temperature probes. For the turbidity meter, that is mainly measures the cloudiness of the water. And total solids, dissolved solids, suspended solids by the gravitational method, by involving the drying and weighing the samples. So some amount of sewage sample is taken, for example 10 ml, and then you dry it for about 101 degrees temperature, and then keep it outside, allow it to the room temperature, again you weigh it, and there will be like before and after. So how much amount of water is being evaporated, and what is the amount of mixture left over. So that is how we will be knowing that solids, suspended solids and the dissolved solids in the water sample. And then the pH. So pH monitoring is very necessary during the analytical purposes. And mainly for this also a pH meter will be available, where this can also be done in a in situ manner. All right, so directly the pH probe, the temperature probe, the COD probe, all these also directly it can be taken to the site location and measured directly, and then taken the samples. And then coming to the chemical analysis for the BOD, particularly there will be a biological measure measuring the amount of oxygen consumed by the microorganisms in the decomposing organic matter. While COD, it is a chemical method where the total amount of oxygen required to oxidize the organic and inorganic substances. For the nutrient analysis and all, for BOD, there will be like method for the bottle there will be a bottle method where the bottle will be enclosed and kept for the incubation purposes, and after certain period of time how much amount of organic matter is degraded by doing the titration method and all, like we will be coming to know. And same the chemical oxygen demand also, where we will be doing by the titration method, or there is a method for COD where the amount of oxygen required also we will be doing through this process. For the nutrient analysis, particularly the nitrogen and phosphorus, the spectrophotometric methods will be used, which mainly determine the concentrations of ammonia, nitrate, nitrite, phosphates. Iron chromatography can also be used for this purpose. And where the values we will be knowing against the blank, and that is how there will be a formula through which the obtained values will be put into the formula and get the results, and that is how we will be knowing the nitrogen and phosphorus content in the sample by placing those samples in the spectrophotometer at a particular wavelength, that is be it 220 nanometers or 340 or 420 nanometers. So there is a different wavelength range where it will be adjusted, and then accordingly the after the after the chemical process and all, we will be putting under the spectrophotometer, and then we will be getting the results out of it. And heavy metal analysis also, like we will be getting through directly, like from atomic absorption spectroscopy, ICP-OES, ICP-MS, and then organic contaminant analysis such as GCMS and LCMS, mainly to identify the pollutants such as pesticides, pharmaceutical, industrial chemicals as such. And then total organic carbon. This also uses the TOC analyzer, where the use of oxidation methods will be determined to get the total amount of organic carbon present in the water. All right. Then the biological analysis, such as the microbiological analysis, where the culturing techniques are used to isolate and identify the bacteria, viruses, and other microorganisms. And fecal coliform and E. coli counts are also used as an indicators of the fecal contamination. Where culturing techniques, as we all know, the plating techniques, what we have, they were after doing several times of serial dilution technique, and then the water will be streaked or spread onto the petri plates with nutrient media, and then with the help of the nutrient media, after certain time or period of incubation, the bacteria growth will be observed, and the yeast, fungi, microalgae, whatever it is, that will be grown after a period of time, and that particular one isolate is formed. We will be taken to the microscope, and we will be checking the shape and structure of it, and then finally we will be sending it to the DNA isolation, and then further identification of it to know what bacteria is playing a major role in this kind of project or this kind of purpose, or dominant bacteria present in this kind of sample and all. And then coliform, that is coliform count we will be knowing, which is mainly the fecal contamination source. And PCR is also like another advanced technique which is mainly used for the detection of the specific pathogens. And I hope we all remember about the corona, where coronavirus was detected by the PCR sampling. And water-based epidemiology. So these days also, like several researchers are focusing on this epidemiology, where urban sewage and all, everything is getting collected at one point, and then how much, till what extent the drugs are getting mixed up into the sewage and water. So that and all, the epidemiology we will be studying through the process of PCR, in order to detect and also to quantify the specific biological markers in the wastewater, such as the viral RNA. All right. Now we will be looking into the treatment technologies. So till now we have seen what is sewage, what are its main implications, and then why is it important, what all analysis to be done, and all we have seen. What are the instruments are there to analyze these sewage water samples and all we have seen. But now we will be looking into the technology. So what kind of technologies they are present in order to treat the sewage. Mainly these technologies, they have over the period of time they have evolved significantly, and mainly to address the increasing demands for cleaner water and environmental protection. Okay, so first one is always the preliminary treatment. Either it might be like sewage water or the wastewater or any kind of water. Initially the preliminary treatment should be done. So this preliminary treatment mainly involves the screening process, the grit removal process, the flow equalization tank will also be there, and the fat and grease removal will always be there. All right. So the first screening, mainly the screens will be used, like bar screens or mesh screens, depending upon the size of the particles or not. So initially, the bar screens will be present where the huge amount of large debris, will be collected. Debris such as rags, logs, sticks, plastics, and such kind of big materials will be screened on or before entering to the next stage of process. But whereas like mesh screens, the pore size is also very less, such that sunlight like little bit, instead of logs and all, they will be like a small amount of paper cuttings, or be it the slightly larger particles, not much larger. When see the plastics, wooden logs, or all these will be screened in the bar screens, whereas the finer particles of the suspended materials will be screened out through this mesh screens. And coming to the grit removal mainly removes like soil, sand, gravel, and other heavy inorganic materials that could actually damage the equipment if further passed on to the next level. Now, third level is the flow equalization tank. This flow equalization tank is that it balances the fluctuations. It avoids the damages of the equipment with its maximum flow. So during this equalization tank, what happens is that the water is allowed to settle down, okay, to be stagnant at a point where the flow can be controlled. All right, so the flow can be controlled, and there should be a consistent treatment. All right, so once the flow is controlled, the fat and grease can be removed. So once the fog that can be clogged in the pipes and all, that has to be removed in the preliminary stage itself, such that it will not clog the pipes or interfere with other treatment processes further. All right, so these are the preliminary treatment process. And coming to the secondary treatment, where initially the sedimentation process will be gone undergone. So initially we have removed the larger debris, then the finer particles and the floating materials, all these are removed. Now we will use the sedimentation method. So sedimentation mainly by using the gravity in order to settle the suspended solids in the primary settling tanks from forming a primary sludge. So this process mainly removes a significant portion of the suspended solids and the organic matter. All right. So if you see in the pictures, first one is the preliminary treatment where the screening has been done after the input of the influent wastewater, and then after removing the larger debris, then it will later go to the grit chamber where it removes the sand and all, and sand and all will be settling at the bottom, and that will be disposed. And then further the skimming of time where the oil and grease of fog materials and all will be released. All right, during the process also there will be like equalization tank where there will be stable flow of water. And coming to the primary one, where the water sedimentation, what we can see here, scum is again nothing but the oil, grease, and all such kind of things will be floating on the surface of water, whereas further separate treatment should be done for that. And coming to the sludge, where coagulants or flocculants are being added to this process, where the suspended particles will collide with each other, forming a flock, and ultimately, due to the weightage of it, it settles at the bottom, ultimately causing formation of sludge. All right, this sludge is heavy in size, settles at the bottom, and then slowly this sludge can be collected and reused for the aeration purposes, or further can be treated and send it back to the environment in the form of fertilizer. and the treated water will be sending it to the next level of treatment. Coming to the secondary treatment process, secondary treatment consists of several advanced process, particularly like activated sludge process and other process. We will be slowly looking into it. And the first one is the activated sludge process. Activated sludge process. Here itself, we will be understanding that various microorganisms will be used in order to break down the organic matter, in mainly in the aeration tank, or else there will be a big aeration tank. After the flow sedimentation and all this process, the water will be sent into this aeration tank where different types of aerators are used. So as we have already seen in our previous classes, where are different types of aeration we have used, the diffused aeration or submerged aerations. So there are different type of aeration tanks used in different locations. If it is industry, one kind of aeration will be used, or in the lagoons or recreational activities, so other kind of aerators will be used. So these kind of, it depends upon the site and all, the aeration tanks will be used. And these in this aeration tank will be quite big, and motors are also will be there in order to for the paddle wheels to rotate and all. And here the microorganism, since more amount of aeration is going on, more amount of oxygen carbon dioxide gas exchange system is going on in near to the water, and hence there is more amount of biological activity going on. Biological activity, as in the microorganisms which are present in the water, will be feeding on the substances of organic matter. That is how the breakdown of organic matter occurs, and air is pumped into the tank to promote the microbial growth, since the microalgae or the bacteria need more amount of oxygen for their survival, whereas microalgae needs carbon dioxide for its survival and growth. All right, so these bacteria can feed on microorganisms, microalgae as well. So there is an interconnectedness between the microorganisms and interdependency, particularly if we call. And then the aeration provides air and promotes the growth of the microalgae microorganisms, which results in the biological flock and then separated using a settling tank. All right, now the basic principles of this is the biological oxidation process, where this process uses this aerobic microorganisms in order to decompose the organic pollutants present in the wastewater. And these microorganisms also consume this organic matter as food, converting into the carbon dioxide, water, and the microbial cells. And that is how, like, most of the researchers here and laboratories are focusing on mostly the biological oxidation process, which is a very sustainable, environmental friendly process, and more amount of uh, byproduct can be obtained through the process and all. So that is the reason, like, more of the researches is going on by this uh, biological oxidation process. Coming into the flock floculation formation, where the microorganisms grow, they clump together, forming a flock, which is essential masses of bacteria and microalgae mixed with other solids and all. And these flocks becomes the ability has the ability to settle down at the bottom of the water, and allows for the separation of the treated water from the biological solids. So this treated water is again going into the next level of treatment. And then the sludge, whatever present in the form of flocks, that is mainly we call it as activated sludge, since the microorganisms played a major role in clumping together. All right, now the sludge, whatever is obtained, is again recycled back to the aeration tank, where in the aeration tank there is more amount of bacteria or more amount of microorganisms are required for the treatment process. So that is how, like, once the collected sludge is present, that is again recycled back to the aeration tank, and in order to maintain a high concentration of microorganism, ensuring efficient treatment. All right. And the key components and the process are the aeration tank particularly, which is very very crucial for the water treatment, and where aeration mixes the wastewater with that of sludge, activated sludge, and aerated. All right. So the air and oxygen is supplied to the tank to support the aerobic microorganisms. The microorganisms break down the organic matter during this stage. All right. Now we will be seeing into the secondary clarifier or the secondary settling tank. Here in this tank, what happens is that the mixture from the aeration tank flows into the clarifier. From the aeration tank, proper aeration is given sufficiently. It has been performing, and then the water from aeration tank is then pumped into the um, settling tank. All right, or secondary clarifier, and then sludge, whatever is settled at the bottom of the clarifier, is, and then it is sent back to the uh, send back to the aeration process, and maintain a minimal microbial population. Now another one is the waste activated sludge. So all these three are interconnected, all these three are interconnected, and excess sludge whatever is present is produced as a byproduct of a treatment process. Yes, after the preliminary stage, a primary stage of treatment, coming to the secondary stage, already all the process will be performed, and sludge will be performed as a byproduct of the treatment process, and which can be removed from the system as waste activated sludge, and this sludge undergoes further treatment as well. All right, if you look into the advantages and disadvantages, advantages are it provides this activated sludge process provides effective removal of organic matter, relatively high treatment capacity when compared to other conventional treatment process, and adaptable to various wastewater types. And the major disadvantages are it requires energy for aeration, and it requires careful monitoring and control monitoring system, and can be sensitive to the variations in the water composition, and also it produces it produces sludge that needs to be further processed. So in this picture, as we see, there are some air diffusers, that is there are some bubbles here, and the water from aeration tank is getting into the secondary clarifier, where the suspended particles are allowed to settle at the bottom. The sludge is collected at the bottom, and which is mainly called as an activated sludge, which is further returned back to the aeration process as an activated sludge, and enhances the performance of the microorganisms for the treatment. And further, the effluent is sent into the next sewage analysis process. This activated sludge, when is fundamentally the same, and it's evolved into various configurations and to optimize the performance, and for the better performance of it, so extended aeration is performed, and extended aeration as in, like, everywhere this activated sludge process is nothing but uh, the collected sludge will be again sent to the aeration process. All right. So this is called as extension aeration, where the longer aeration time is required, where the treatments this is a modification of it, where modification from the previous one, where the larger aeration tank will be there, And aeration tank also, like the longer the time as well, that the more amount of aeration, the more amount of good activity will be going on of microorganisms, more treatment and all. These are the other configurations, and this leading to the complex breakdown of organic matter, and more stable sludge can be obtained. So this is often used in a smaller communities or a packaged plants due to its simplicity and availability, ability to handle the fluctuations in water wastewater flow. And the second one is the oxidation ditches. These oxidation ditches are modified activated sludge process used for the biological wastewater treatment. Alright, it mainly operates as an extended aeration system with a continuously looped flow, and it provides the effective organic matter removal, nitrification, and denitrification process. It is widely used in the municipal and the industrial wastewater treatment plants due to its low operational cost, energy efficiency, and in simple in maintenance. And these are the components of the oxidation. And if we look into the picture here towards your right, it is the oxidation ditch method, where this circulation tank is there, that is called as an oxidation disc tank, ditch tank, where it can also be called as a raceway tank. And as we can see the flow pattern here, or else there is a continuous flow of water in a loop manner, and where there is will be a peristaltic pump here, where with along with the paddles, such that the aeration can also be provided along with the circulation of it, and the proper mixing of nutrients or proper mixing of the contaminants will be provided, where the wastewater once introduced into this oxidation ditch, the water will be circulated, more amount of aeration is provided, more amount of treatment will be occurred, more active microorganisms take place, and then the clear water will be then sent into the secondary clarifier, where it is allowed to settle at the bottom, where the sludge is being collected, and the surface water whatever is floating in that will be sent into the disinfection facility. Disinfection, as we have seen in our previous slides, by the usage of ozone or by the usage of UV facility, chlorination. So depending upon the type of disinfectant available, and depending upon the type of discharge or reuse of this water and all, the type of the disinfection method will be used, and further it will be discharged for either for irrigation purposes or any other purposes for its reuse. Whereas the sludge collected at the bottom of the clarifier will be again used for the sludge return during the aeration process, or it can be used and collected in the other treatment process for the sludge thickener tank, which will be for later for sludge will be thickened and sludge will be dried and made it into powder, and water is all removed and then made it into the powder, and again it can yet it is, since it is having more amount of nutrients even after treatment process and all, it is used for drying, and the powder can be used for the environmental applications for the good growth of plants, and improve the fertility of the soil. Coming to the combination or components of an oxidation ditch, one is the looped channel. So a closed channel, a closed loop or a raceway shaped basin that facilitates the continuous flow of water. The depth could be maximum of 3.5 meters, and the retention time will be total of 12 to 36 hours. And the aeration uses the mechanical aerators, that is the surface rotors or refuers or paddles, anything to provide the oxygen and keep the solids in suspension mode. So the type of aerators will be the brush aerators, surface disc aerators, diffused air systems, etc. So these are the aerators used for this purposes. And secondly, clarifier. So here in this, a separate settling tank allows the sludge to settle. All right, and the return activated sludge is recycled back to the ditch in order to maintain the microbial population. Coming to the next one will be the sludge handling system, where WAS is periodically removed in order to prevent the excessive biomass accumulation. The next one is the we will be seeing about the working principle of an oxidation ditch. All right. So in the step one, the influent. Okay, the influent enters the ditch. Okay, then it will be rotated in a looped channel where it mixes with along with the activated sludge. And in the second stage, the aeration will be provided and more amount of biological treatment occurs. And these mechanical aerators provide the oxygen to support the microbial community. And these microorganisms break down the organic pollutants, nitrogen and phosphorus for its growth. And nitrification and denitrification process occurs by reducing the nitrogen levels. And in the third step, the clarification, that is the settling and the separation process. The treated wastewater flows into a secondary clarifier where the solids settle, and the clear effluent will be discharged for further treatment. That is the chlorination, UV disinfection, anything, chlorination, whatever. Here, the fourth step will be the sludge recycling and wasting, that is return activated sludge, that is RAS, where some amount of settled sludge is returned to the oxidation rich and aerated. And waste to activated sludge, that is WAS, where excess sludge is removed for disposal or further processing. And these are the main advantages and disadvantages and applications of this oxidation ditches. That is, advantages are the high treatment efficiency, that is, it removes BOD, COD, nitrogen, and phosphorus. Low operation cost, environmental friendly, and energy efficient due to the extended aeration. And stable performance. So handles variable wastewater loads effectively with minimal sludge production and no primary sedimentation required. Coming to the disadvantages of the oxidation ditch, where a large land area is required, and energy consumption for aeration is more, and odor issues are slightly higher, and skilled operation is necessary in order to control and monitor the system. Coming to the applications of this oxidation ditch, it is mainly useful for the municipal wastewater treatment, industrial wastewater treatment, decentralized and advanced nutrient removal and treatment systems. All right. So we will be now looking into the sequence batch reactors. These three come under the advanced oxidation process, where first one is the extended aeration, second one is the oxidation ditches, and the third one is the sequencing batch reactors. All right. So, what are the sequence batch reactors? So, in a sequence, it will occur, and in a batch mode, the name itself says. All right. So, let us see about this. The sequence batch reactor is a fill and draw activated sludge system used for the wastewater treatment. And this batch reactor operates in a batch mode, treating the wastewater in a single tank through the sequential cycles. And it is widely used in the municipal and industrial and decentralized wastewater treatment plants for its due to its flexibility and efficiency. The components of SBR system are typically it consists of a reactor basin, where a single unit for all the treatment processes will occur, that is biological sedimentation, decanting, and all these, there will be a single unit for all this treatment process. And aeration system, fine bubble diffusers, the fine bubble diffusers or mechanical air creators provide the oxygen for the biological treatment, that is the aeration system. And mixing system, where the agitators or the diffusers, all right, keep the solids suspended and enhance the biological activity. And the decanter system, the floating or fixed decanter removes treated effluent without disturbing settled sludge. All right. And the control system, this control system mainly it is programmable, logic controller PLC, which is mainly automatic cycle phases, will be recorded by this. Alright, so if we see into the working principle of SBR, alright, then it occurs in a this whole SBR process occurs in a single tank, alright, following the fixed cycle operations. So in the phase one react, that is the aeration phase. Uh, Aeration phase, where it is provided aeration is provided for the biological degradation and of micro algae, sorry sorry microorganisms or any organic matter, and then the nutrient removal, and these microorganisms consume the organic pollutants, nitrogen and phosphorus, and the third step will be the settle, like the sedimentation tank, and the decant. Decant is nothing but the removal phase, where the whole water is can be removed without disturbing the settled sludge. Idle, that is the sludge wasting and the reset phase. Excess sludge is removed from the bottom of the tank. The tank is prepared for the next cycle, and each cycle lasts from 4 to 8 hours, depending on the wastewater characteristics. So, as we see here, first is the anoxic fill, okay, without any contaminations and all. Then we will be seeing the aerated hill. So aerator, the diffused aerator or whatever it is, it will be like continuously stirred such that there will be like a good amount of air transported into the water. And the third one is the react, the where the reactor, where the clogs will be like there present, and then the clogs will be settled here, and then the decant water at the top water will be taken up, and again the anoxic. So this is a cyclic process where everything is like interconnected and all. So the design parameters of this SBR mainly includes the hydraulic retention time, that is 4 to 24 hours, solids retention timing, that is 10 to 40 days, and cycle time is 4 to 8 hours, and organic loading rate is like around um, zero point 0.2 to 1.2 kg per BOD. All right, and then aeration method by using this diffuse method, and the effluent quality. The effluent quality is majorly like shows the effluent properties or the characteristics of wastewater, and that should be less than below 10. All right, the, the main advantages of this SPR system is the compact design, cost effective, efficient organic and natural resources, and removal process, nutrient removal, sorry. Efficient organic and natural removal process, and flexible operation, automated control. And disadvantages are mainly like require skilled operation, batch mode limitation, foaming and sludge bulking issues, which lowers the shell life of the equipment, and then higher energy demand, all these. So coming to the applications of SSSBR, that is the municipal wastewater treatment, industrial wastewater treatment, decentralized treatment process, and nutrient removal systems. So after the activated sludge process, now we will be looking into the trickling filters. All right. Yeah. So coming back, the second one after the activated sludge process is the trickling filters. These are well established biological wastewater treatment technology which utilizes a fixed bed of media in order to remove the organic matter from the wastewater. So how does this trickling filters work? By the formation of biofilm. So the biofilm formation is there where water is initially distributed over the media of bed of media, which can be rocks, gravel, or synthetic materials. The microorganisms establish themselves on the surface of the media, forming a biological film, right? And then organic matter removal, whereas the wastewater trickles down through the media, the microorganisms in the biofilm consume the organic matter, breaking it down, and air circulation within the filter provides the oxygen needed for this aerobic process. And as the biofilm grows thicker, parts of it is slowed off and then carried away with the treated wastewater, and the slowed material is then typically removed in a secondary clarifier. So, do you understand the process? How does it work? By initial formation of biofilm, and then the filtration, minimal filtration will be done, and the formation of the biofilm also will be done by this. And then secondary, like water, organic matter is removed by breaking down the organic matter with the help of microorganisms, biofilm consumption, and air circulation should be proper within the filter media such that it provides the oxygen needed for the aerobic process. And slowing is as the biofilm grows thicker, it should be slowed off or taken away, and as it is clogging the material to pass from the media. And these are the main key components, that is the filter media, distribution system. The filter media mainly provides a surface for biofilm growth, and it can be natural materials, either it could be rock or gravel or any plastic material. And the distribution system will be mainly the sprays, or it also distributes the wastewater evenly over the filter media. It often uses rotatory distributors. Under drain system is another one where the treated water is getting collected and biofilm is lowered, and it also allows the air circulation process. And the main advantages of this process is that it is relatively simple in operation, low in energy consumption, and it is very robust in operation. And the main disadvantages are the odor potential is there, and fly nonsense, and space requirement is very big, and potential for clogging. So the main application of these trickling filters are that it are mainly useful for the treating domestic and some of the industrial wastewater process, and they also often used in a small to medium sized communities. And major variations are that trickling filters can also be clarified as a high rate or low rate, depending upon the hydraulic and organic load rates. And the recirculation can be used in order to improve the treatment efficiency. And is, in essence, the trickling filters provide a reliable and more effective biological wastewater treatment method, particularly suitable for the applications where simplicity and the lower energy consumptions are very important. So these are the variations for this. And so as we look into the picture here, so there is a sprinkler on the top, and there is a layer here where it will be like biofilm will be here. And so this is the filter media where the water is sprinkled and it has to pass from the top to the bottom, and there is a collection here where the effluent channel, we call it as, that can be used for the other purposes. And this is a feed pipe, this is the filter media bed, and air is also pumped inside. And filter supporting bed is also present where the biofilm formation is present. And if we look into the other picture here, so this is actually the industrial one and the design of it. And this is the rotating influent distributor where the water is sprinkled from the through the filter media bed, and the air is provided here in order to give bubbles and aeration to the microorganisms. And also the air vent is also present, and then filter media here. And The clarifier is here, and the water once treated can be used in this type of pump here. So now that we understood the trickling filters, now we'll try to understand the rotating biological contractors. The among the treatment process, all right, among the secondary treatment process, first one is the activator sludge process, second one is the trickling filters, and the third one is the rotating biological contractors. Alright, so rotating, as the name itself suggests that it is like rotating. By through rotation, air is getting circulated, and it is a unique approach for the biological pollutant removal process. And the main principles of it is the attached growth. So the RBCs utilize a fixed film biological process, and these microorganisms grow and attach to a solid surface. Either it could be a rotating disc or any plastic surface present or any material for the biofilm to get binded, and then it grows on it. So this constraints with activator sludge, where the microorganisms are suspended in the wastewater. Aerobic treatment. Aerobic treatment, as in the rotation of the disc, extremely expose. The rotation of the disc alternatively um, exposes the biofilm to the wastewater and air, providing the necessary oxygen for aerobic biological biodegradation and organic pollutants. So the water will be circulated, the air will be circulated, and then the removal of water is very easy, and biodegradation also will be provided by this process. So how do they work? So this is the industrial one where we can see this, and formation of disk capacity. Yeah. So they actually work like the disk is there, right? So the disk capacity for its rotation. So the more the disk rotation, there will be more amount of circulation of water. So there are series of spaced disks, typically made up of plastic, are mounted on the horizontal shaft. Then the shaft rotates. So this is the shaft, and this kindly rotates, and partially submerging the disc in the water. So the water level might be this much, and then the disc are allowed to submerge the disc into the water, and then the biofilm formation, where the microorganisms attach to the disc surfaces, forming a biofilm. As the disc rotates, the biofilm comes in contact with the disc, and then and the wastewater, absorbing majority amount of organic matter. And coming to the aeration, where the disc rotate out of the wastewater, the biofilm is exposed to air, and allowing for the oxygen uptake. Alright, and then slowing and clarification. Excess biofilms loss of the disc and is carried away with the treated water. A nearby clarifier is then used to separate the slotted solids from the treated effluent. All right. So do we understand now? So how do they work? They have a the disc rotation, and the shaft rotates and partially submitting the water into submerging the disc into the water, and then there will be a formation of biofilm, where the microorganisms attach to the disc surfaces, forming a biofilm, and it comes in contact with the wastewater and absorbing organic matter. And then the as and when the disc rotates, the aeration occurs, and following the oxygen uptake. And slowing and clarification, once it provides gets into huge size, that will be taken off, and the secondary clarifier is then used to separate the slotted solids from the treated effluents. So mainly, if we see into this RBC's advantages, it is relatively simple in operation, low energy consumption, robustness, and good for smaller systems, and they are well suited for the smaller communities or the decentralized systems. And coming to the disadvantages, it is very sensitive to the climate, maintenance is difficult, space requirements are more, and applications are mainly for the secondary treatment for municipal and the industrial wastewater, and they are mainly effective for removing organic matter, and they can also achieve the nitrification process. And if we see here in this rotating biological filters, so here is the wastewater, where the wastewater consists of COD, chemical oxygen demand, that is the organic component, the ammonium, nitrogen, total nitrogen, turbidity. So it is pumped into the rotational RBCs, where it is sent through the peristaltic pump, and where the disc can be rotated at a speed, at a certain speed. Hydraulic retention time and standard retention time will be operated in order to have a good mixture and the treatment of wastewater. And then the water is sent to the settling tank, where the waste sludge is again collected for the further processes, and the effluent is collected, and this clear effluent can be again reused for the environmental purposes or fertilizers and all this, recharging the water and all. So coming to the fourth one, which is the advanced tertiary treatment. So this advanced tertiary treatment is the one, like also comes under the secondary treatment process after the advanced sludge process. Then the rotatory biological filters, and then another one, and this is the fourth one actually, where it is advanced treatment process. So in this process, where nutrient removal majorly takes place. So nutrient removal in the wastewater can is an essential for protecting the aquatic ecosystems, and excess nutrients, particularly the nitrogen and phosphorus, can be which majorly leads to the eutrophication of the nearby water body, can be actually dealt by this process, and causes the excessive algal growth. So the presence of nutrients are the feed for the microalgae for its survival, and yet these microalgae have the several benefits as well. These days, extensive research is also going on by providing the wastewater treatment by giving it to algae, because algae takes up the nutrients for its growth and survival. And then once it is the water is treated, it is harvested, and the sludge can be collected and dried, and the powder of it, we can obtain the good amount of protein sources there, good amount of carbohydrates, fuel, you can get the biofuel, and then carbohydrates, proteins, and then biochar also what we can get, which is ultimately useful for several purposes. The protein which can be used for the fish aquatic purposes, fish feed, and the fertilizer, the components also are present in the biochar. Such like that, several positives are there for this algal growth, and there is also like oxygen depletion and harm to the aquatic life as well. So with the excess of nutrients, there is a disadvantages of having the depletion of oxygen, ultimately leading to the death of the aquatic fishes. So coming to the nitrogen removal, where nitrification process plays a major role. So there is an aerobic process where ammonia is then converted to the nitrite and then to the nitrate by the specialized bacteria, that is the nitrosomonas and the nitrobacteria, where this process is a crucial step in the nitrogen cycle, and it also plays a significant role in the wastewater treatment, soil fertility, and the environmental management. So in every process for the conversion of ammonium to the nitrate, also there will be ammonium monooxygenase enzyme takes place, and with the help of nitrosomonas species, it is then converted to the nitrate, and with the help of nitrate oxidoreductase enzyme, with the help of nitrobacter species also, the nitrate is then converted to the nitrate process. So this is the overall nitrification reaction. And coming to the denitrification process, where this is an anoxic process, where the dissolved oxygen is absent, and nitrate, after the nitrification process, there is a formation of nitrate. From the nitrate, again it is then converted to the nitrogen by the specific bacteria. All right, so there is a process during this process, the bacteria plays a major role, where this nitrogen gas is then released into the atmosphere. This process requires a carbon source. All right, carbon source, either it could be glucose, it could be acetic acid, or it could be any other carbon agent like glucose. Starch also. Starch is again like nitrogen compound, but yeah, glucose, acetic acid, propionic acid, these are all different types of carbonic acids, and provides the carbon source for the microalgae to grow. And this process is an essential step in the nitrogen cycle, and also plays a key role in the wastewater treatment, agriculture, environmental management, by removing the excess of nitrogen from the ecosystems. All right, so if you look into this, where the dissolved oxygen is lacking in this system, but during the denitrification process, air diffusion, nitrification, and oxygen levels varies during the nitrification denitrification process. And the third process is the anamox, where the anaerobic ammonium oxidation takes place. And in this process, where ammonia and nitrate are directly converted to the nitrogen gas under the anaerobic conditions, which is a relatively new process and more energy and efficient process than the traditional denitrification and nitrification process. And it is also performed by anamox bacteria, and highly efficient in the removing of nitrogen and from the wastewater. So anamox is a process where ammonia and nitrate are directly converted into the nitrogen gas under anaerobic conditions. It is a new process, energy efficient, and highly efficient in removal of nitrogen and wastewater. Here in this, nitrifying bacteria and anamox bacteria plays a major role in conversion of ammonia to the nitrogen gas. So coming to the phosphorus removal, that is and all what we have discussed is the nitrogen. Now if we look into the phosphorus removal, it can be done by two processes. One is a biological phosphorus removal, and the other one is a chemical phosphorus removal. So the biological phosphorus removal, where phosphate accumulating organisms, particularly these kind of microorganisms, take up the phosphates, and that release and then taken up. Take and then uptake the phosphorus under alternating anaerobic and aerobic conditions. And the enhanced biological phosphorus removal process is a common form of removal of phosphorus. Whereas in the chemical phosphorus removal, it is highly difficult, and by adding the chemicals to it, and it is not economically friendly as well because of the addition of the chemicals, and it mainly involves the adding of chemicals such as the aluminium, iron salts, and to precipitate the phosphorus as a solid. No, it precipitates with another form of metal or any salt and forms a product. All right, so that is how it is done, and these solids, they are then removed through the sedimentation or the filtration process. So if you look into this biological phosphorus removal, where there will be the aeration basins, where they with the existing with the presence of the existing clarifiers and all, the water can be removed, and the fermentation is done, the aerobic process, and that is how like the microorganisms plays a major role in this process. Whereas during the chemical process, it is with any metals, iron metals, aluminium, ferrous, like iron, whatever it is, it combines with it and forms another product, which is ultimately settled down, so sediments at the bottom, precipitated, and then gets precipitated, and that is how it can be removed in the form of sedimentation or filtration process. So the main key considerations for this tertiary removal process is the combined process. Often the nutrient removal systems, where the nutrient such as both nitrogen and phosphorus both can be removed by the process of biological and chemical process in order to achieve the optimal results. And the process optimization could be by doing the varying the pH, varying the temperature, varying the dissolved oxygen levels, and the carbon source availability, which are mainly critical for the optimizing the nutrient removal process. And further, the environmental regulations always regulations, where the stringent regulations will be driving the need for the more effective removal of nutrient technologies. And the sustainability always takes a major role, and the resource recovery of the nutrients is always becoming more important during this process. And the technologies used are the mainly the membrane bioreactors, sequencing batch reactors, constructed wetlands, and activated sludge system with modified configurations, that that is the BNR systems and all, biological nitrogen removal systems particularly. All right. So if we now look into the membrane bioreactors. All right. So the membrane bioreactors represent a significant advancement in the wastewater treatment technology. They combine the treatment with the membrane filtration, which results in the high quality effluent with a smaller footprint when compared to the conventional system. And these membrane systems are the filtration process mainly, which involves the microfiltration or ultrafiltration with a biological treatment process, typically an activated sludge system. This is the combination which allows the separation of solids and liquids using the membranes and eliminating the need of secondary clarifiers. So, how do they even work? The wastewater enters into the biological reactor where the microorganisms break down the organic pollutants, and which is similar to that of activated sludge process. So instead of settling the solids in the clarifier, the mixture is then filtered through the membranes, which retain the soils and bacteria, producing a high and clear quality effluent. And this membrane acts as a very fine filter, preventing the suspended solids, bacteria, and other viruses from passing through it. So membranes can be either zero point 0.1 micrometer, 0.05 micrometer, or zero point 0.2 micrometer, zero point 0.5 micrometer. So depending upon the purpose of it. And then the advantages of these membrane reactors are the high quality effluent, smaller footprint, high biomass concentration, improved nutrient removal. And the disadvantages include this membrane falling. Yeah, frequently the membrane has to be replaced, and the higher energy consumption is required because membranes directly don't work on its own. Some pump should be there where the due to the pressure pump, the membrane acts to the vacuum, the water gets passed through the membrane. So higher capital and operational costs are there, and applications are mainly they are suitable for the applications requiring high quality effluent such as the water reuse and sensitive environmental discharges, and they are mainly used in locations with very where space is limited. All right. And mainly in the laboratories, if we can say, yeah, we can use this membrane filtration units. And the key considerations are the membrane type, depending upon the pore sizes and availability, depending upon the application of it, and depending upon the different membrane materials can be used. And the falling control strategies for preventing and controlling the membrane falling are essential for successful MBR operation. That is a regular cleaning of the membrane or passing of water in order to clear the clogs of the membrane. So that kind of things can be done, or backwashing, that kind of things. And the maintenance by regularly membrane cleaning and maintenance is required. So all right, so this is the membrane bioreactors what we see here. This is the anoxic tanks where the conditions are pressure. There is a pressure gauge here. The membrane separation tanks are also present where the water is initially sent into it, and through the pressure gauge the water is sent through the membranes. And water, the clear water is then collected from the outlet of it. So the inlet will be here, and the membrane and the water, see how it is going on. So finally, once the water is getting passed through the membranes, the water is again obtained as very clear, and can be used for several purposes. So that is how the membrane bioreactors work. And coming to the advanced oxidation process. So this advanced oxidation process, and so these are all the disinfection process and all. This will be dealing in the next class. So I hope you all are following my classes, and kindly go through the previous classes what I have told. So there are several sedimentation and aeration process, and disinfection process what we have already studied in the earlier classes. So once if you go through the other slides as well, you will be aware of all the classes what we have done. And that's all for today. And in the next classes again we will be dealing with more treatment technologies and wastewater treatment, the sludge removal, sludge treatment, all these we will be discussing. And thank you for your cooperation. Thank you all, and that's it for today. Thank you.